It is indeed a privilege to have amongst us today Dr. Viraj Suvarna, Medical Director, Boehringer Ingelheim India Private Limited. Sir is the Head of Medical Affairs and Research, Pfizer Incorporated, and the and the Executive Committee member of Indian Society for Clinical Research.
Sir pursued his MBBS from the reputed Topiwala National Medical College, Mumbai, and MD in Pharmacology from Grant Medical College, Mumbai. Sir has also completed his MSc in Pharmaceutical Medicine from Hibernia University, Dublin. I kindly request Dr. Suvarna to come on the stage and address the audience. Yes.
Hello. First of all, I would like to thank Dr. Anil Pandit, Dr. Parag Rishi Pathak, and Devjit Hazra for inviting me and ensuring that I come here safely and on time. It is an honor to speak here. I've heard a lot about symbiosis. In fact, I wanted to do a distance learning MBA from symbiosis, but there's absolutely no time, but I'll still try to do it. Although now I will be 51 in a few days from now. This topic, plagiarism in clinical trials, when Dr. Anil told me to speak on it, first what I told him was plagiarism, of course, is very common, but in clinical trials, maybe not as common as with publications, with presentations. So even today when I present to you, who knows, some of the slides I may have taken from some colleagues or maybe from the internet, but as long as I have referenced them and made it very clear that these are not my thoughts but the intellectual property of someone else, and more importantly taken permission from that individual, then it is not plagiarism. Then of course there's plagiarism in the way we do our dissertations, our thesis. Nowadays there's so much available on the internet. People do a lot of this copy paste or whatever, and luckily there is a tool apparently where people can subject your document, and that tool searches the internet and tries to see whether there is some amount of plagiarism. Does anybody know the etymology of this word, in the sense this word plagiarism has come from, as usual, Latin or Greek, and what does it mean? It means kidnapping. So you're kind of being a kidnapper because you are taking somebody else's intellectual property and trying to show it as your own. So that is something which is obviously not right. And today I was actually coming from Panchgani to Pune by that Nita Volvo bus, and I was sitting next to a guy who asked me, what was my topic about. I told him, and then he asked me, but what is wrong with copying? So then I had to explain to him, and it kind of helped me even in preparing for this presentation. Where do I... यहाँ से कर सकता हूँ? Okay. I think you'll have to put it on slideshow. Yeah. So the first 4 slides will be just to reinforce the fact that plagiarism is not right. So it's essentially the appropriation of another person's ideas, processes, results or words without giving appropriate credit, including those obtained through confidential review. Imagine, you are a peer reviewer in some journal, someone else's manuscript comes to you, you go through it, and then you appropriate from there into an article that later on you send to another journal and get it published. And this has happened, I've read it. So imagine, even that can happen through confidential review of others' research proposals and manuscripts. Presenting the data or interpretations of others without crediting them and thereby gaining for yourself the rewards earned by others is theft, and it eliminates the motivation of working scientists to generate new data and interpretation. See, in some colleges, even to get promotion, you need to have lots of publications. So I have even seen sometimes students, when they do their PG dissertation, naturally at the end of it they will acknowledge maybe their head of department, guide, etc. But sometimes what I've seen is there are people who actually take all these different manuscripts and put it together as if it's their credit, and that helps them in promotions. Plus, in the industry, what I've seen is, see, nowadays there's this practice of asking certain key opinion leaders or external experts to come and speak for you, either at an advisory board meeting or at a speaker development program or even at a continuing medical education program. Now, these speakers, they get tiered, tier 1, tier 2, tier 3, and based on that, the honorarium is decided, the fair market value. So naturally, these doctors also know that the more publications I have, maybe I get more money for that talk as compared to someone else. So because of these reasons, sometimes all this fabrication basically happens. And the difference now between fabrication and falsification. Fabrication is you make up data or results, and of course the recording or reporting them when actually nothing is there. Falsification is you're manipulating research materials, equipment, processes, or changing, omitting data or results such that the research is not accurately represented in the research record. And the most dangerous of all falsehoods is a slightly distorted truth. It's like in medicine, the difference between, say, an illusion and a hallucination. So typically we say, optical illusion is a mirage in the desert. Actually, that's not an illusion. It's a hallucination because there's nothing there and yet you're seeing something. Illusion is like you see maybe a rope in the dark and you think it's a snake. So there is something there, but you are seeing it differently. So similarly here, the difference between fabrication, where it's something where nothing exists and you've made it up, and falsification, where there is something, but you know that maybe a negative trial or a negative report or manuscript or publication will not even be published in some journals, which is wrong. Nowadays, people do publish negative results. But because sometimes the journal also wants only positive stuff because they feel the readers will want only positive stuff, but that is not right. We need to publish everything, including negative results, because let's understand that just because something is negative, means that study is negative, it doesn't mean that the study has no value. In fact, the study has a lot of value because then you understand what went wrong here. And then you read the paper, you build on the limitations, you do another study, and eventually you advance medicine. But if you didn't know, it means all negative studies don't get published. Then what happens is the evidence that we talk of is no longer evidence-based medicine, it's evidence-biased medicine. And what's the difference between based and biased? The letter I. And if you look at the I, so ultimately it is your trained eye. If that can discern, can separate out the wheat from the chaff, that is when a reader will really be able to decide as to what is original, what is genuine, and what is not. It's not that easy. Plagiarism is a serious offense that could lead to paper rejection, academic charges, and termination of employment. It will seriously affect your scientific reputation. Don't do it. Unacceptable paraphrasing, even with correct citation, is considered plagiarism. So what I was told when I had to write my first manuscript, my boss told me, see, to write anything, it could be just a review article, it could be a systematic review including a meta-analysis, but how you go about doing it is, obviously you'll have to read many articles. After reading, and of course it has to be targeted reading based on what is it that you want to communicate to the audience. After reading, you close your eyes and now you think in terms of what do you want to write, and then you write. Now, as you're writing, some part of what you're writing could be taken from this article or that article, reference it. But don't take the entire sentence from there and put it here. I know some people do it and then they put inverted commas and they reference it, but that is also not really correct, though some people say up to 10% is fine. But point is, don't directly lift and put. You read a lot. Based on all that reading, you decide what you want to write, and it is your thought, your intellectual property that you're putting on that paper. But you reference that, obviously I read all these different articles from where I've taken certain points. But at the same time, you've not lifted the paragraph from that other person's article ad libitum. So that is important. So now I come to the presentation per se, which the first part, obviously the practice of taking someone else's work or ideas and passing them off as one's own. It generally happens, as I said, with publications, presentations, thesis. Do you all know the difference between a thesis and a dissertation? What's the difference? This is what I have read. A dissertation is generally what happens at an MD level, where it's not absolute original work. Other people before you have done similar work. You've read all that. When you read the discussion section of the paper, you obviously go through limitations and then you build on it, and that's how you advance science. So that's a dissertation. What you do at a PhD level is original research work. That is called a thesis. Though there also some people might say, are you sure it's absolutely original? I'm sure some people have done. But there is a difference between a thesis, which is at a PhD level, and a dissertation, which is at a slightly lower level. Then, of course, what Dr. Pandit told me is, if you believe that in clinical trials is not that much plagiarism as, with publications, etc., you can also get into other ways in which people copy. So one is obviously the generic versus the small chemical original product. And similarly, the biosimilar against the original biologic. And there is a difference. In clinical trials, are we saying that people copy study designs? That means the protocol. Protocols are actually published. When you read all these medical journals, nowadays people even publish the entire design. Why do they publish it? They want people to know exactly how they did that study, so that one is you can reproduce it, you can test whether what they're saying is correct or no. Secondly, you can build on it. You can identify maybe certain limitations, and then when you do your study, you can do it even better. And that is the way, as I said, medicine or science advances. So I don't think that would be called plagiarism. Then, of course, how the study is conducted. Now here again, people actually talk about how their study was conducted, because they would like others also to follow the same kind of quality, the rigor with which they conduct. Of course, you can do it even better, because there is no end to learning, as I think one of the speakers said even in the other room. And in India, apparently, this is not true, but people believe copyright means right to copy. So that I will come to a little later, where, see, I am not against generics. I will tell you both sides of the story, and I will leave you to decide as to what is right and what is not right. This is actually here, I only can be called guilty of plagiarism, because this is a slide which I've taken from my colleague Dr. Avijit Hazra, who is a DM in clinical pharmacology at Calcutta, IPGMER. So I should have at least written at the bottom of the slide, acknowledgement, because he has obviously given me permission to use it, but since I'm speaking on plagiarism in clinical trials, obviously I should put the acknowledgement, Dr. Avijit Hazra. But basically, just to tell you what happens during a peer review process, and that's why so many of us are so scared to even send an article to a journal. But actually, even if it gets rejected, especially these foreign journals, they give you a very nice reasoning as to why it was rejected. So you learn a lot. And then obviously you can build on it. And then the next time you send your next manuscript, this manuscript, you can't now send it if they've rejected it, but you learn a lot. But the next time you write, maybe it will be much better. Maybe you, another manuscript, send it to the same journal, and then who knows, they might actually accept it. Then, of course, you get extremely upset when it gets rejected. So it's like, you quack, we whack. So here perhaps the peer reviewers actually made out, quack as in you have these doctors who are quacks. So you have also quacked as in you have taken material from elsewhere and then tried to show it as your own. In fact, I was shocked that people even do it for thesis, dissertations. I did my MD from JJ. JJ is a whole library, whole building as a library, and the first floor, so many theses are kept in some cupboard. And I could actually hear some people saying, we'll take one, thesis from there, and then they just make some change. And it's horrible, if that is actually true. Then, of course, who can have conflict of interest? Now, this is where I was trying to tell you that it's important that people who are reviewing it don't have any conflict of interest. Otherwise, as I mentioned earlier, sometimes it has happened that a reviewer has appropriated some sections of a manuscript that he had to review, and then he passed it off as his own in another manuscript, which is the worst thing to do. Then, of course, there are duplicate publications. So just because you want, that publish or perish kind of culture, you want as many publications to your name in your CV, because that somehow makes a difference. So they, submit one manuscript to one journal, and then they make few changes there and then submit it to the next journal. So that becomes a duplicate publication. It has to be totally different. Like, for example, you can submit maybe the global results of that study, and then you can publish other things like subgroup analyses and all. That's okay. But you can't have duplicate publications. You can't have salami publications where people just slice and dice and then publish in different journals. Again, just two. And there are journals, called predatory or pseudo journals, which accept just for a fee. Whether they do peer review or no, also we don't know. So like that we can do. But as I was telling you earlier, negative results nowadays get published. There's a whole journal of negative publications. Plus, even for industry, it's important. That's why you have data steering committees, data safety monitoring boards. They take the decision to publish, even if it is a negative study. So sometimes, at interim analyses, they will decide whether I need to prematurely terminate the study, either because of a safety issue or an efficacy issue, or even a futility reason. That means even if I continue this study till the end, I don't think I will get any difference between the two drugs in terms of the way those curves are separating. So sometimes I may prematurely terminate, and then I have to publish. Because nowadays, when you even want to do a study and you get regulatory approval, you need to post that study on a website, ct.gov, even in India, ctri.nic.in. So later on, if you don't publish, people who go on such sites can ask you, and you have to publish. So what I'm trying to tell you is, in the past, yes, negative studies were not getting published. Nowadays, negative studies do get published. But still, there could be some cases where they don't, or they take very long to get published. So this we've already seen. The only difference is misrepresentation is fabrication or falsification, and misappropriation is plagiarism. So these are two misses that you should keep away from. Again, if you want to know the definition, the act of appropriating and using ideas, writings, or work of another person is one's own without giving credit to the person who created the work. So as long as you give credit, and ideally even take that person's permission, and preferably don't lift it, that is the best way of doing it. Then, as I said, citations let your reader know that you're using someone else's ideas or words. Proper citation is an important tool to avoid plagiarism. Then apparently there is a site which can help you to know whether this is plagiarized or no. And that's what, when in doubt, cite. Now I thought, after all this plagiarism bit, let me first come to what is a clinical trial, and then try and talk a little bit about what I said earlier about a generic and a biosimilar. So this is, I found, the best definition by Sir Austin Bradford Hill, who was involved in the 1948 trial, the British Medical Council on streptomycin and TB. So what he says is, it is an ethically and scientifically justifiable experiment in human beings with the aim of answering a precisely framed question, which means don't confuse yourself with too many questions to be answered. So that's why this primary endpoint is most important. So that is your hypothesis. Of course, you can have secondary, tertiary endpoints also, but your study is powered for the primary endpoint. The sample size is calculated based on whatever you want to answer in terms of that null hypothesis. And based on that, you decide what is your sample size. So don't try to answer too many questions. Though, of course, you will have subgroups, you will have secondary endpoints. That will give you an indicator, but then all you have to do is do another study. But this time, that subgroup or that secondary endpoint becomes your primary endpoint. Then you can definitely claim much more than from a secondary endpoint or subgroup. Now, this is in general what any drug, obviously you need to first discover or invent the drug. Everyone knows the difference, no? Discovery and invention. Discovery is, it's just there, and maybe sometimes serendipitously you discover it. Invention is where you actually, it's not there and you have to invent it, you have to make it. But then it goes through, of course you'll apply for a patent and things like that, but then it goes through animal tests. Nowadays there's reductionist, so tissue, cells. People even use the computer, so in silico, computer-aided drug development. And then you come to human beings. So phase one is obviously in normal healthy volunteers, except when you have these life-threatening conditions like cancer or AIDS, where such patients are included in phase 1 because we don't know how long will they live. Then, of course, it moves on to phase 2. So once you know from animal studies, you do what is known as allometric scaling. You come to a particular dose for human beings, and then you first test for safety in human beings. Then you move normal human beings, and then you move on to patients. So that's phase 2, smaller groups. Then once you see, that yes, it works, safety as well as efficacy, then you want to obviously test it on larger populations. So then you move to phase 3, and that is when it's even not just site, but multi-country. And eventually, then you submit that data to the regulator, get approval. But even after that, you may want to do studies even post-approval. So that's in a phase 4 setting. Now, PMS and phase 4 are not exactly the same, in the sense post-marketing surveillance is obviously in the phase 4 setting. But all phase 4 studies need not be post-marketing surveillance. You can do a randomized controlled clinical trial even in a phase 4 setting. Phase 4 only means that phase after you get marketing approval. Post-marketing surveillance means you're only surveying, you're not intervening. It's non-interventional in a naturalistic setting in the real world, the way doctors anyway would use that product. You're just observing, capturing information, preferably per label, especially if you're from the industry. If you're from academia, you can even collect off-label information. And what now people are saying is, can we do hybrid designs? So try and mix up the two. Because see, in a randomized controlled clinical trial, you are taking patients, very carefully you're selecting them, inclusion, exclusion criteria, so that you get a homogeneous patient population where your drug has the best chance of showing that it works, which means, is it efficacious? But once you get the approval and you start using it in the market, you use it in all kinds of patients. So that is what you test as effectiveness. So an efficacy and an effectiveness setting are not the same. In the real world, you can't exclude any patient. In the real world, the patient pays for the drug, so may not be compliant. And you're not selecting the patient so well as compared to pre-marketing. So now what people are saying is, can we do hybrid designs, large, simple trials? And then what I say by GCRP and GCP is, GCRP is good clinical research practice, GCP is good clinical practice, what you do day to day. And ideally, doctors need to be good at both. So if you participate in GCRP clinical trials, it doesn't have to be global, it could be Indian also, then you also become attuned to a particular way of standardized, uniform treatment per protocol, documentation being very important, informed consent, etc. And then once you come to the real world also, some part of it may overlap and it may improve your practice. Plus, as a good practitioner, your clinical acumen can also be extremely useful if you want to provide inputs to sponsors into the way they need to design their clinical trials to be more clinically relevant, to be more pragmatic. So that's why from evidence-based practice to practice-based evidence, and you can do a lot of research in practice also. I know many Indian doctors will tell me, have you seen how many patients I see? I don't have the time, but by and large, I feel if you really want to do it, you will take out some time. I know abroad, you work from Monday to Thursday, Friday is only devoted to research. Fine. So that's why they can churn out more papers, etc. But in India also, we actually see so many patients that if we really hone our observational powers, we will be able to see patterns, and then we will design or formulate a research hypothesis and then actually think in terms of how to now test this hypothesis in practice. Of course, we will need time, people, a whole team and things like that, but it can be done. It's not that it can't be done. See this. What Sir Richard Doll is saying is, every practicing physician conducts clinical trials daily. As he's seeing patients, every time a doctor treats a patient, he is performing an experiment, because when you're treating the patient, do you know what will be the outcome in that patient? You actually don't know at that point in time. That's why it's better to say a doctor only treats and God cures or God heals. We are only tools in his hands. Now, in that process, we are actually doing a trial. So why don't we document it formally? Because I know in that hierarchy of evidence, experience is right at the bottom. But that experience, if well captured, can provide a lot of information to doctors, because there are limitations of even RCTs. So nowadays people are trying to do both RCTs and real world evidence in the right way. so that they can complement each other. Because otherwise, an RCT can be extrapolated only to the kind of patients who are in that RCT, only to the kind of setting in that RCT, which is not really there in your practice. They are two worlds apart. But yet doctors use that data, interpret, and then extrapolate to their individual patients in their practice. So obviously there will be some gap. The N of one RCT, or randomized controlled clinical trial, is actually the highest in that hierarchy of evidence, but very difficult to do. Imagine in practice, as a doctor, you are blinded, patient is blinded, and then you test two different drugs in that same patient, but sequentially. And then you come to know to which drug does this patient respond the best. It's not about saying this drug is better than that drug. It is about finding out this patient responds best to which drug, this or that. But industry will never do such trials. Ideally, we should do, because it's not that my drug is better than another person's drug. In fact, Dr. Rohini Kelkar knows in febrile neutropenia, there's a guy called Dr. Jean Klastersky, a medical oncologist. In an editorial in NEJM on febrile neutropenia, he actually concluded by saying the real question is not about whether, for example, liposomal amphotericin B is better than voriconazole. It's not that. This kind of patient perhaps would respond better to this drug. Another kind of, and it's not just a drug, it could be a regimen, could be a combination, could be the sequence. That is important. And industry needs to come together in what I call a spirit of competitive collaboration to facilitate pragmatic clinical trials that answer clinically relevant questions, not whether one drug is better than another drug. This is just to tell us, we all know the double-blind and sometimes double-dummy trial. A gold standard clinical trial will always be prospective, randomized, which means every patient in that trial has an equal chance of being allocated to either treatment A or treatment B. Means you're trying to reduce bias to the extent possible. So prospective, randomized, double-blind, active or placebo controlled clinical trial with a focus on outcomes, hard endpoints, mortality, morbidity, not just does it reduce blood pressure better or does it get HbA1c to goal better. Those are softer surrogate endpoints. This is just to tell us the cost of bringing a drug to market, and this was way back, I think, in 2003, when they said that the total cost is around $897 million, of which the actual cost is about $400 million. Then, of course, there's this post-approval R&D cost, which is basically your post-marketing surveillance and plus maybe some new uses for that drug you want to explore. But this is important. Opportunity cost for capital employed. See, the same money, instead of putting it into a pharmaceutical company, if I put it elsewhere, I will get much better returns more reliably as compared to if I put it into a pharmaceutical company, because although we call it discovery, I call it risk-covery. So much of risk. And typically when you invest, high risk, high returns, not in the pharma industry. Although I know there are some examples like Lipitor, $13 billion, blockbuster product from Pfizer, Humira from Abbott, more than even $13 billion. So there are a few such examples. But in contrast, there are many, many examples where industry pours in so much of money. I'll tell you now, the cost has gone up to, this is on an average 2.87. Some people have even pegged it at $4 billion. So you spend so much, and what is your return? Is it really predictable? Not really. And once the drug comes to market, then also something can go wrong, like some really serious adverse event which could not have been picked up during pre-marketing trials, and that's the reason we need to do post-marketing surveillance. And then the drug is out. And once the drug is out, it's not just that. So many patients will sue the company, legal suits, and the company loses money and also credibility, image, trust. How do you determine price? This is important because you need to understand why is a generic able to price itself so low, and why is it that a company which does innovation unfortunately has to price it a little higher, at least till the patent. Once the patent expires, the price drops. So there's a difference between these terms, cost, price, and value. Cost is, you saw, $2.87 billion US on an average. Price is determined not by R&D. So what I just told you, cost, that is sunk cost. So how they determine price? By something known as the anticipated income stream from that potential blockbuster. We don't even know whether it will be a blockbuster. And that in turn is dependent on three factors. One is your period of exclusivity. Now you think I have a patent 20 years, nobody can copy. But that patent you get in the very beginning, when you first had that, maybe the molecular structure or something of that drug, and you had some idea that yes, it will. So you apply for a patent. At that time, you have yet to develop the drug. You get a patent, 20 years, but you take 10 to 12. In the past, it used to take 15 years to even get that drug to market. Then you might say, no, but I still have 8 years. But within 1 year of you coming to market, other companies, which are also innovators, will come with a similar product, not your product, but similar. Like with anticoagulation, we came out with a factor 2A inhibitor, a direct thrombin inhibitor. Other companies immediately came with factor 10A inhibitors within a year or two of launch. So our exclusivity shrunk from 8 years to just 1 or 2 years. So period of exclusivity is not necessarily a long period. Differentiability. How different is your product from others in the market? Now you may think, oh, pharmacologically, no. But from a clinical perspective, does it really make sense for a doctor to shift from what he's currently using to your product? What does it offer? Is it clinically significant? Safety, efficacy, convenience, etc. And third is the market sustainability. Is the market willing to pay at that price? So people do pricing research, value, volume, etc. And eventually, based on maybe competitor pricing, a certain price is determined. But what is most important is value. That means what do I really, if I am the buyer, see as the value I'm getting? Means for this much of cost, what is the value or the outcome or the benefit that I'm getting from that product? And this is in terms of where all that money goes into. And you will see that manufacturing is only 8%. So this is the reason if I just copy someone else's, obviously I may not use the same process because there could be a patent on process as well. So I use another process because there are different processes, and that's why it's called reverse engineering. You come to the same product, it won't be the same, but it's more or less the same. And all that I need to do is manufacture it. I don't invest at all. And I only need to do a bioequivalent study. So I take 12 volunteers, healthy, 12 another. Give one set the original, the other set the generic, and I look at concentration versus time curves if they are superimposable within certain limits. It's approvable. And that too in India only in the first 4 years. After 4 years, it's an old drug. You don't even have to do that. Just go to a state FDA. Just show some chemical or pharmaceutical equivalence, and that's it. Now you tell me, obviously the price will be low. So that's all I'm saying. I'm not saying generics are not good. But if that generic is not of the same quality, then you have a problem. Because different generics have different prices. Why? I don't know. All of them have to only manufacture. But the originator, yes, has to do a lot more. And the data, the clinical data has come only from the originator, which then they copy. But this is again not called plagiarism because per the law, for a small chemical and a generic, you don't even need to have clinical or therapeutic equivalence. It's only chemical or pharmaceutical equivalence and pharmacokinetic or bioequivalence. Clinical or therapeutic is not expected, even abroad, because people want to reduce the prices of drugs. So if you have enjoyed your exclusivity for 20 years, for example, obviously now generics should come in. So in the US, both generics and the innovator both do well. This is just a quote. Just as the price of a textbook is not determined by the cost of the paper of its pages, and the cost of surgery has little to do with the price of the surgeon's scalpel. Why do we pay him? For his expertise, for his qualification, training, years of experience, and because of which now his error rates are very low. That's why we pay. The most expensive product is the one which does not work, because if it does not work, then you will have all kinds of problems. So sometimes the question is not whether you can afford to prescribe the more expensive product, but whether you can afford not to prescribe. And this is just one kind of, 1 minute, different definition of the word health. I know typically WHO defines it as, how does WHO define health? So that is the usual definition. This is a different kind of definition. It's an attitude. Means it's putting onus on the individual also. It's an attitude of well-being that when nourished by a sense of personal responsibility from within and a coordinated support system from without, enables a person to live life to the fullest. Health should be a want and not just a need. So if you want to be healthy, then you will adopt healthy lifestyle practices. You cannot smoke, drink, do all kinds of other things, sedentary life, and have this big paunch, and then expect that I should not have an MI, and then if I have an MI, it should be very cheap and things like that. No. So sometimes I feel it could be an incentive for you to stay healthy and to stay out of hospitals and out of reach of doctors, because then if you don't do that, then you end up losing a lot of money. This is just a study we did on finding out the burden of stroke. So on the one hand, you have very cheap warfarin, which is given to patients of atrial fibrillation to prevent stroke. Warfarin is so cheap, yet patients don't take it for other reasons. It has problems. And then they get a stroke. And that stroke can cost them 5 lakhs just for that one episode which happens, and just for that 3 months or 1 year. But whole life he may be living with that stroke. So you can imagine how much he's... Oh, sorry. 10 minutes, huh? Okay. Sorry. This is just an example. I won't take you through this, but just to tell you that on the one hand, you have a very cheap amphotericin B. On the other hand, you have a more expensive voriconazole. But the voriconazole, if it is more effective and it's safer, ultimately, when you look at the overall cost, say for the amphotericin B, it comes to 17,150, but for the voriconazole also it's around the same. Although to start with, the voriconazole, if it is from Pfizer, say the original patent holder VFend, it will be more expensive. But in the long run, ultimate differences could be more or less the same, and the more expensive product could actually turn out to be cheaper in the long run. But now if you ask me to compare a VFend with a voriconazole generic from say Sun or something, then this comparison will not apply. But there, of course, what we need to show is that the two, the generic and the original, are equivalent from an outcome perspective. Then you can do what is called as a cost minimization analysis. Nobody knows because there is no data. Like I'll tell you this one study called PROBE. I won't take you through the details of it, just to tell you that in Pfizer we had this product called latanoprost sodium for intraocular hypertension or glaucoma. So these are drops you put and then the pressure goes down. Now naturally another Indian company, Sun, came out with a kind of a generic latanoprost. Now doctors from Shankar Nethralaya, which is a very reputed ophthalm institute in Chennai, they decided to do this, and a small crossover kind of design. That means when you do crossover, the same patient initially was given say Xalatan, and then after a washout period is given latoprost. So within the same patient you can check for differences, and they actually found differences. Now normally we say generic and original, no difference clinically or therapeutically, but sometimes these things happen, and this is just one example. I won't go through the entire design because I don't have time, but just to tell you that significantly more reduction in intraocular pressure was seen with Xalatan, whereas with latoprost the intraocular pressure actually, instead of coming down, went up. So obviously the difference was statistically significant. This is just to tell us that with biosimilars there's a slight difference because it's a biologic which is more complicated or complex. The process is the product, and that process is proprietary. It cannot be shared. So naturally you will take another process. You will start with a different cell bank. Could be Streptomyces cerevisiae, Saccharomyces cerevisiae, or there are other like Pichia pastoris for this hepatitis B vaccine, and you can have different sources, but ultimately it's not enough to just show quality or manufacturing specification differences. You must also show clinically whether they are similar. So that's the only difference with a biosimilar and a biologic. But in India, again, the rules are a little more lax as compared to the European Union or the US. So Indian biosimilar manufacturers may find it difficult to export to these countries, but in India, of course, they can definitely do it much faster. So with a biosimilar, you need to be careful about interchangeability, even pharmacovigilance when you're reporting adverse events with brand name rather than generic name. And when you do these bioequivalent studies, it's not only enough to determine whether these are prescribable, but also whether these are switchable. That means this guy is well controlled on this originator product. Now you come in with that cheap generic or biosimilar. Can I shift him? Will his condition be well controlled? This is just to tell us, we keep talking of a human genome project, but the genetic variation in the way people respond to drugs, the difference is not that much between continents. The difference could be within India, but not that much between India and Americans. We always feel, Americans are much more, bulkier, so you can have higher doses for them. And with Indians, we start at 1.25 mg rather than the 10 mg, say for Ramipril. Then again, this thing about industry funding and non-industry funding. So there was actually a study done which found that the quality of studies done by industry is better than quality done by academia, because as I said, it's not only done by industry, there are external factors also there, a statistician from outside. Even sometimes industry outsources the study to academic research organizations, and then industry doesn't have any control over the way that study will be run. So please understand that just because you have industry, it doesn't mean always that it's negative. This is my last slide, and this is what even Ayurveda tells us. It is much more important to know what sort of a patient has a disease than what sort of disease a patient has. Because individually, all of us are heterogeneous in the way we experience the same disease and in the way we respond to a drug. Even in homeopathy, people actually label the individual as sulfur or phosphorus or whatever, and then decide the treatment, which particular tincture. So that's all I had in my presentation. I know I have gone above your limit, but please feel free to ask questions till 5:15. Dr. Rohini Kelkar has to speak, but any questions? The topic was beyond plagiarism. Huh? I started with plagiarism, but then what Dr. Anil told me is there are so many doctors here and they have all kinds of questions. So you even deal with other typical industry, how you have doctor bashing, hospital bashing, now you have industry bashing also. So yeah.
So you mentioned about unacceptable paraphrasing as a plagiarism. So can we differentiate between acceptable paraphrasing and what is unacceptable?
So acceptable paraphrasing, as I told you, is you close your eyes, think about what you want to say, put it there. See, paraphrasing is, suppose you ask me a question. I need to first paraphrase what you've asked and send it back to you so that then you understand whether I have understood your question in the way you wanted me to understand. No? That's paraphrasing. So similarly here, if you put your own intellectual property and paraphrase, it's fine. But if you take a whole paragraph only from that, then first of all, it's not paraphrasing itself. But the point is, do not lift ad libitum from what somebody else has written. Read it and then put it in your own words. So that's why I'm saying the best is you read, then you keep all that aside. Close your eyes, think in terms of what do I want to write. And then when you realize that this was taken from that article, reference. But it will look different. It won't look the same as that article. So then it is your article.
Because I am a Wikipedia editor, and in Wikipedia they say that paraphrasing is the key not to get into copyright issues.
Because in paraphrasing, what you will do is you will put it in your own words. You will not distort the content or what exactly that person was trying to say. You will put it in your own words. So therefore, no one can then claim that you are lifting. But unacceptable paraphrasing is you want to paraphrase, but you are still taking words from there. So when you read it, you will realize.
Do you know of anybody or any instance truly that anybody in India has suffered academic punishments because of, is it actually there? We keep hearing academic punishments.
You're
Has
right.
it actually occurred?
I don't know about India. It may happen in India also, I don't know.
Abroad
but
also.
Abroad, Eugene Braunwald, who is the person who, people read that textbook, Cardiology, he was once involved in an act where his resident did something which was unacceptable, some form of plagiarism, while doing a clinical trial. Now, naturally, he cannot say it was my resident and I didn't know, because ultimately you're the boss and that person is your PG student. So he had to take the blame, and he had some censure. In fact, in all these presentations on GCRP, you will find one example of Eugene Braunwald, because Eugene Braunwald everyone knows. And if he also can be pulled up, so you can imagine. And abroad, even investigators who are caught not doing research or falsifying, fabricating, etc., there is a blacklist. So then and that's posted on the US FDA website. So immediately companies also know, don't go to these investigators. Now similarly in India, we should have that blacklist. And it should be all public. Everything should be public. Even if I pay a doctor for a legitimate service advisory board, it should be. Like abroad, the Sunshine Act ensures that doctors who are paid by companies, it's there on the company website. So even for the doctor, when he's doing his income tax and all, he has to be careful. Sure.
I have seen
I know, and later on, I have seen that the same guy, then all his articles have been redacted because it was found out. Like in Pfizer, it happened on Lyrica, pregabalin. This guy, Pfizer used to give what are called as grants for investigators to do investigator-initiated research. Now, in that, we cannot monitor what he does. We just give the funding, and then it's his study. So he did it, and in good faith, we thought he's doing good work. And it got published. We even used it in promotional materials on Lyrica. And then when it was caught, and there is one Indian also, Dr. R.B. Pandit or something in center of, I think, Madhya Pradesh, who has been pulled up for this kind of falsification.